Thanks, Mike. So the last speaker of this really great symposium is the other co-organizer, Forrest White. So Forrest is an associate professor in the Department of Biological Engineering here at MIT, and he has a large degree of expertise in mass spectrometry, and probably the best person for measuring phosphotyrosine and phosphoserine in network models. So Forrest,
Thanks.
without further ado.
All right, so thanks to Mike for inviting me to give the talk. Hey, Mike, do you have that laser pointer still, or is, oh, never mind, got it. All right, cool, yep. All right, so I know I'm between you guys and booze, so I'll try and be quick here. And I actually don't have any really complex pathway diagrams, which is kind of strange. But anyway, so what I wanted to talk about today was really this question that's been sort of bothering me is how do we get from genetic mutations to therapeutic targets? And as I think all of you know, there's a lot of work going on in basically TCGA and mapping out cancer genomes. And what's been identified are a lot of mutations that people have historically seen, like P53 or PTEN, and in ovarian cancer, again, P53, and in breast cancer, a lot of the same mutations that people have sort of expected, as well as some novel mutations. But by and large, most of these mutations aren't necessarily druggable, and it's not clear how you would treat patients that have these particular mutations. And so to gain insight on that, basically we're interested in the systemic effects of these genetic mutations. And so if you think about what happens when you have like a PTEN mutation, for instance, that gets transcribed and translated as a non-functional protein that then alters signaling networks, as we all understand. But then downstream of those signaling networks, you get phosphorylation of transcription factors and global transcription that then basically changes splicing and microRNAs. And then that leads to proteomic changes on a global scale that then all feeds back into these altered signaling networks. And it's basically a feed-forward loop. The system continues to progress over time. And in the context of a second hit, of course, you get transformation. And what we'd like to do is basically understand this system and understand how we can therefore treat these tumors that have these particular mutations. Another way of thinking about this is simply to say that genetic mutations lead to complex effects on cell signaling networks. And if we know these networks, we can actually know the biology underlying these networks. We can intercept and destroy cancer cells. And importantly, this same concept is not just applicable to cancer, but basically to any complex disease. So the particular story I want to talk about today is this question of how do RAS mutations alter substrate ERK substrate utilization, or sort of larger put, do RAS mutations alter ERK substrate utilization? And this is sort of if we take a step back, a different question that I'd like to address is basically when we take cells and stimulate them with EGF, the cells basically activate ERK and undergo proliferation. We take the same cells and stimulate them with IGF-1, for instance, the cells will start to migrate, or PDGF, the cells will start to migrate. They're still activating ERK. So is ERK always hitting the same substrates under those conditions? And are the other signals therefore controlling the behavior of the cell? Or does ERK basically use different substrates under different conditions? And so to address this, we basically have to be able to map ERK substrates. And to do this, we turn to technology that was developed by Keyvan Shokat. And back in 1997, Keyvan developed these AS kinases, where he took the ATP binding pocket and bumped out the gatekeeper residue in the ATP binding pocket, such that these analog-sensitive kinases were able to use an analog ATP with a bulky side chain on it. And so in this latest approach, basically they take these AS kinases with this, I'll just call it ASATP because it's easier, this ASATP, and it's a thiophosphorylated ASATP, such that the AS kinase can use the thiophosphorylated ASATP to thiophosphorylate protein substrates. And these thiophosphorylated protein substrates can then be alkylated, and then he generated an antibody that recognizes this alkylated thiophosphate group. Or they can be digested to peptides, solid phase captured by iodoacetyl beads, and then released with an oxidizing agent as phosphopeptides that could then be analyzed by mass spec. And the nice thing about this is that we're actually pretty good at analyzing phosphopeptides by mass spec. So if we can get to this stage, we should be able to identify substrates of any given kinase that's been bumped out and that's where we've used this ASATP. And so that was sort of the premise when Scott Carlson, a graduate student, joined my lab a few years ago. And I said, hey, Scott, why don't we use this approach? It's published. How hard could it be? We'll just go in and map out ERK substrates. And then determine how RAS mutations affect these ERK substrates. And that would be sort of the first 6 months of his PhD. And then we'd figure out something else. And so after about 9 months of work on the protocol, Scott actually got it to work. And basically got it to work by altering multiple steps in the protocol, including a critical step of changing the pH here that basically selects for the thiophospho instead of just thiopeptides. And then a final step, which is cleanup by IMAC, which is immobilized metal affinity chromatography, to enrich for phosphopeptides and get rid of nonspecific binding at this step. And so throughout this protocol, Scott did the analysis, and then at the end of the analysis, he ends up with these detected known substrates, which is really good because these are sort of the positive control. And then down here, we have all these potentially novel substrates, which is awesome because this is basically new ERK biology that we could discover. And the way that we define these substrates is that they had to have a proline in the +1 position because that's sort of the canonical motif for ERK. And they had to be phosphorylated, of course, because coming through the analysis as phosphopeptides, and therefore they should be ERK substrates. So everything looked good until we got to this protein down here called ATF7. And ATF7 is a transcription factor, and there's a couple of papers that specifically say that it's not an ERK substrate, but is instead a P38 or a junk substrate. And so that raised this question of, all right, if that's not an ERK substrate, at least by these other papers that have done in vitro kinase reactions, how many of these other proteins are really not ERK substrates? And so that raised a further question of basically how specific is this ASATP use for AS kinases? And so Scott took these fibroblasts and stably transfected them with either the AS-ERK2 or wild-type ERK2, stimulated them with EGF, lysed them in kinase buffer, and did the kinase reaction with the ASATP to get these labeled substrates, and then alkylated the substrates and did a Western blot with this antibody against the alkyl thiophospho group. And basically what we see is, or what we should see is basically a lot of bands in the AS-ERK2 expressing fibroblast and no bands in the wild-type ERK2 expressing fibroblast because these have no AS kinase. Unfortunately, we do see bands here, and if we expose the gels for longer, you can see there's actually quite a few bands that come up in these cells that do not have an AS kinase. And so there's two possibilities here. One is that there's other kinases in the cell that can use ASATP, and that's actually not that surprising when you think about it because proteins have sort of different conformations and they're not rigid structures. So maybe there are some proteins for which ASATP can fit into the pocket. The other possibility is that that thiophospho group is being cut off of ASATP and added onto regular ATP, and then the cells are just the kinases are using regular ATP that is now thiophosphorylated. We actually don't know the mechanism behind what's happening here, except that this is a pretty big issue for us because this gives us a lot of non-specificity in our assay. So to address this question of non-specificity and ASATP use by non-AS kinases, we turned to SILAC. And so Scott took the AS-ERK2 expressing fibroblasts, grew them up in heavy media that has heavy arginine and heavy lysine, took the wild-type ERK expressing fibroblasts, grew them up in light media, activated ERK, lysed the cells, did the same kinase reaction, mixed them together. And at the end, what we should be seeing is that for background signals, we should have equivalent light and heavy peptides, whereas for ERK2 substrates, they should be specifically enriched in the heavy peptides compared to the light peptides, basically in those cells with the AS-ERK2 versus the wild-type ERK2. So now if we go in and look at the data, FGFR substrate 2, this is a peptide that had a proline in the +1 position. It was phosphorylated, looked like an ERK substrate. There's the heavy, there's the light. It's obviously not an ERK substrate. ETS2 repressor factor 2, this is a very well characterized ERK substrate. There's the heavy and there's the light, in agreement with what we would expect. CDC42 effector protein 1, a novel ERK substrate. There's the heavy and there's no light there. Again, a novel ERK substrate. And so through this analysis and 6 biological replicates and some statistics, we end up with basically 80 substrates, including 67 novel substrates of the ERK2 MAP kinase. that cover actually many different downstream pathways and many different aspects of cell biology. And what's pretty cool is when you look at the literature, ERK basically does everything. So there's 5,000 papers on ERK, and it's related to almost every process you can imagine. Most of the substrates in these processes aren't very well characterized. And in fact, ERK is very well known to drive migration. How exactly it drives migration isn't well characterized, but here's a suite of Rho GTPase proteins that are all ERK substrates according to our analysis. And so we basically get proteins in cell cycle and apoptosis, which is good, and transcriptional regulators, and a variety of other processes. The ones that are in red here are actually very well characterized, are previously characterized ERK substrates. And then we went in and looked at some of these in particular. And so the first thing that when we sent in this paper, one of the things the reviewers asked us to do is basically confirm that these were ERK substrates through in vitro kinase assays. Unfortunately, we had already done an in vitro kinase assay because that's how we got to this point. But to satisfy the reviewers, we still made recombinant ERK and recombinant proteins and did in vitro kinase assays for a handful of them. And they all worked, of course. The other thing we did is to do some bioinformatics and show that the substrates, our putative substrates, I should say, are all enriched in D domains and DEF domains, or are enriched in D domains and DEF domains relative to the mouse proteome. They also cover a broad variety of different components in the cell, at least according to their Go annotation. So one of the proteins that was particularly interesting to us is this protein called ETV3, which is an ETS family transcription factor. There's only like 7 papers or so on ETV3, but one of the papers describes it as a transcriptional repressor. So it binds to DNA and represses transcription. And there's another paper that says it's not an ERK substrate, but in our hands it actually is an ERK substrate. And the other reason that it was very interesting to us is that it actually was non-canonical. And in other words, the site that we saw had a proline in the -2 position, but not a proline in the +1 position. It was the only site that came out of the analysis that that lacked a proline in that +1 position. So Scott basically HA tagged ETV3 and then dropped it into the cells. And if we stimulate the cells with PMA, which activates PKC and therefore activates ERK, we get a nice gel shift up here. If you inhibit MEK with UO126, you get a gel shift down here. Scott did this experiment. I still don't understand why he did the experiment, but it came out kind of cool. Basically, where he mixed the antagonist and the agonist together on the same cells, and he ends up with basically this multiple banding pattern, potentially indicating multiple phosphorylation sites on the protein. If we go in and map ERK phosphorylation sites, or ERK-dependent phosphorylation sites on ETB3 by mass spec, we end up with 8 ERK-dependent phosphorylation sites, which are all shown here. And so now the question is, okay, so it's multiply phosphorylated, and many of these sites are ERK-dependent, but what does phosphorylation actually do? And since it's a transcription factor, one assay you can do is basically just ChIP-seq. So chromatin IP followed by sequencing to see where this protein is bound on DNA. If we inhibit MEK with UO126 and block phosphorylation of ETV3, this protein binds to and represses around 3,000 or so genes. If we then activate the cells with PMA, which then leads to increased phosphorylation on ETV3, we lose binding to DNA completely. And so it either sits down and represses 3,000 genes, or it lifts off the DNA and doesn't bind and doesn't repress these genes. And so then the question came as to how this happens. How does it either bind or unbind from DNA? The first thought was that phosphorylation altered nuclear localization. So we looked at nuclear localization of ETV3, either under UO126 or PMA stimulation conditions. And in both cases, it's nuclear. So then the second thought was, well, maybe it alters protein degradation. Maybe phosphorylation increases, is a signal for degradation of the protein. So then we looked at ETB3 levels following cyclohexamide treatment, and then either with PMA or PMA plus UO126. And in all cases, basically, we don't see a change in stability, but ETB3 sort of turns over with a half-life of a little bit less than 2 hours. So the protein's constantly turning over, and then in response to phosphorylation, it appears to unbind from DNA. And so we end up with this model where ETB3 is sitting there repressing, like I said, 3,000 or so genes, and slowly being turned over. In the presence of ERK activation, we get phosphorylation on 8 sites on ETB3. That lifts it off the DNA, primes it for transcription. And interestingly, one of the genes that's regulated by ETB3 is actually ETB3 itself and DDX20, its requisite cofactor. And so now when ETB3 lifts off of the DNA, you get expression of ETB3 and DDX20, which then, of course, sits back down on the DNA and shuts off this transcription, allowing you to get a wave of transcription for these 3,000 or so genes, depending on the activating signal that's there. One of the other genes that's actually very strongly regulated is DUSP6, which is the phosphatase that dephosphorylates the ERKs. If we do an analysis of these various genes, we get enrichment of these genes for cell cycle and proliferation, as well as NF-kappa B and inflammation-associated genes, indicating potential crosstalk between these networks. So that's basically how we map ERK substrates and one particular ERK substrate. What I realized sort of as we were going along here is that there's 67 novel ERK substrates. Figuring out one of these took us about 2.5 years. And so I'd be really old by the time we get through, even older, I guess, by the time we get through all of these. And we still haven't really addressed that question that I had at the beginning, which is basically, can we figure out the effects of RAS mutations on ERK substrates through this sort of analysis? And so we turn to try and answer this question rather than working through the biology of all of those ERK substrates. And basically, we use this colon cancer DLD1 cell line that has mutant KRAS. Bert Vogelstein's lab has nicely taken out that mutant KRAS through homologous recombination. So we have an isogenic cell line that basically either has mutant KRAS sort of in the normal cells or lacks mutant KRAS in these isogenic cells. And then we can stably transfect those to either have AS-ERK2 or wild-type ERK2. And so in a triple labeling SILAC experiment, we can take the DLD1 cells with wild-type KRAS and AS-ERK2, grow them up in light media, DLD1 cells with mutant KRAS and AS-ERK2, grow them up in heavy media. So now the difference between the heavy and the light is the difference in sort of substrate utilization or substrate phosphorylation in the presence of mutant KRAS. And in the middle channel here, we have DLD1 mutant KRAS cells with wild-type ERK2. So anything that shows up strong in this channel is actually not a substrate, but is sort of background signal. And so through this analysis, we can now quantify ERK substrate phosphorylation. In these cells, we ended up with about 110 ERK2 substrates, about 130 or so phosphorylation sites on these substrates. 30 or 40 of these are novel in the context even of our other studies. So we're up to about 100 novel ERK substrates in these two studies. And interestingly, what we see is actually altered substrate utilization in the presence of, so this is mutant KRAS, divided by wild-type KRAS. And some of the phosphorylation sites are up by as much as 4.5-fold in the presence of mutant KRAS. Interestingly, some of these at the very high end are splicing factors, which might be kind of interesting in terms of the biology of mutant KRAS in these cells. And then we have some phosphorylation sites that are actually decreased in the presence of mutant KRAS. And we have a whole suite of phosphorylation sites in here that are significantly increased. And several of these end up basically being epigenetic regulators. And so we're now trying to understand the role of ERK phosphorylation and increased substrate utilization on regulating the epigenome in the context of, and regulating chromatin in the context of RAS mutations in these cells. So basically, KRAS mutations lead to very significant changes and very complex changes just in ERK substrate utilization. And the question came as to whether we could translate this analysis to the study of genetic mutations on human tumors, because that's where I started all of this. That's exactly what we wanted to work on. My disclaimer is that I actually don't think this is going to work anymore. So I went into this fairly naive, and I thought, well, we'll make these point mutations in PTEN or make these point mutations in RAS, figure out what they're doing. Of course, in any given tumor, there's not just a single mutation, but there's many, many mutations. And if each of these has this level of complexity on the network, it's going to be almost impossible to figure it out. So giving this talk in front of Doug a few months ago, and Doug basically stood up at this point and said, with computation, everything is possible. And it's true. It might be possible eventually. Right now, I'm not sure that this is actually possible. So my sort of second goal in doing this is basically similar to what Mike Cohn presented earlier, basically using mass spec to define altered signaling networks in tumors with the goal of identifying better therapeutic targets. So even if we can't link genetic mutations directly to altered networks, altered signaling networks, and understand the complex biology interlinking these two things, at least we should be able to identify activated networks. And our specific tumor that we've been working on lately is glioblastoma. What I wanted to talk to you about today is a really brief story on glioblastoma tumor tissue xenografts that work done by Hannah Johnson in my lab in collaboration with Jan Sarkaria at the Mayo Clinic. And basically what Jan has done is take human tumors and then grow them up in mice as these tumor xenografts. And in collaboration with Jan, we picked out 8 different tumors based on their EGFR expression. And sort of with the goal of cherry picking tumors to have either EGFR wild type overexpression or EGFR V3, this constitutively active mutant form of EGFR expression. So we picked 2 tumors that have minimal EGFR or EGFR V3, 3 tumors that have pretty high levels of EGFR, and then 3 tumors that have EGFR V3, this constitutively active mutant form. And then we did 4 replicates of each tumor. So in total, 32 tumors, each set of 8 injected into 4 different mice. And so the workflow for what we're doing is basically taking these tumors, homogenizing the tissue, labeling digesting the proteins up into peptides, labeling those with iTrack, and then doing phosphotyrosine profiling with phosphotyrosine enrichment by immunoprecipitation followed by IMAC, and then onto an Orbitrap for basically quantification and identification. We also do protein expression profiling with some minimal amount of fractionation and then analysis on a 5600 instrument. So we put all of this data together and then look at basically how each of these tumors looks for both protein expression and phosphotyrosine signaling. So from the protein expression profile, this is approximately 1,100 proteins quantified. If we just look at this figure on the left-hand side of the screen, one of the things that sort of jumps out at you, although it's fairly pale here, is that each of the tumors has basically different protein expression profiles. And so even at sort of a very gross level of just looking at the 1,000 most abundant or so proteins, we're already seeing quite a bit of difference between these tumors. What Hannah wanted to do is go in and see, are there particular proteins that are enriched in the V3 expressing tumors compared to either the wild type or low EGFR expressing tumors. She identified 63 proteins that were differentially expressed in the V3 expressing tumors. Most of these are upregulated in V3 expression. And if she goes then to the Rembrandt database and looks to see how the mRNA profiles correlate for these various proteins, correlate with patient prognosis, she ends up with 5 proteins whose mRNA profiles correlate with patient prognosis, and the combination of all 5 proteins ends up actually being a pretty good predictor for patient prognosis at the mRNA level. Whether or not this is true at the protein level is something that we'll need to test going forward. So what we were really interested in is the tyrosine phosphorylation data and basically comparing the tyrosine phosphorylation pattern across these different tumors. And this is where the data gets very confusing to me, because as a signaler, I'm used to sort of activating EGFR with EGF and then looking at the downstream proteins. You get increased phosphorylation on EGFR and increased phosphorylation on the downstream proteins, and especially on the adapter proteins right next to EGFR. For these tumors, what we basically see is especially for tumor 15, shown here in the middle, we get strong increased phosphorylation on EGFR itself. But then when we look at SHIC or Sybil or PLC gamma or STAT3 or GAB1, all these adapter proteins that are known to bind to these exact same phosphorylation sites on the receptor, we don't see that increased tyrosine phosphorylation on these proteins. And so it seems like for tumor 15 at least, we're getting this really strong phosphorylation at the receptor, but that signal's not going anywhere. Which is, Joan's face says it all. You guys can't see it, but it's got this confused look on it. And that's exactly sort of my look when we started looking at the data. And the other interesting thing is as we go through this, basically what we're seeing is actually some increased signal on basically some of these proteins downstream of EGFR V3. And so what Hannah wanted to do is see, all right, are there any phosphorylation sites that are actually enriched in the V3 tumors or differentially phosphorylated in the V3 tumors? She ended up pulling up the following phosphorylation sites that were significantly enriched or altered in the V3 tumors. And what's really confusing is just looking at the top 3 here, as well as HRS, which didn't make the slide, but is also significantly enriched in V3. And what's confusing about this is that EGFR V3 is basically supposed to be a weak signaler that is not supposed to activate endocytosis, and yet these proteins all are involved in the endocytic and trafficking machinery, and they're all increased in phosphorylation in the V3 tumors, exactly the opposite of what we expected. So I've been listening to all these talks where everybody has these beautiful mechanisms, and I don't have a mechanism. Like to get to the punchline, I'm still very confused. Unfortunately, as we moved on to human tumors, it gets actually even more confusing. So I still don't have a punchline or a mechanism for this. If we look at the tyrosine phosphorylation networks for these various tumors, what we're hoping to see is basically that we'd see an EGFR signature here, an EGFR V3 signature here, and something different here. What we basically see is 8 different signatures. And if we try and cluster these phosphorylation sites, the clusters basically break out into phosphorylation sites that are increased in one tumor or another tumor. But it basically really begs this question of personalized medicine and basically trying to use this data now, as Mike Koch talked about earlier, to try and identify the activated pathways in each tumor and then identify the optimal therapeutic targets in each tumor. But unfortunately, given the complexity that we see here in these tumor xenografts, as well as in human tumors, in these glioblastoma human tumors, it's not obvious that we're going to have a single drug or even a single couple of drugs that will treat all patients. And so I think we're going to need to use some of this data to actually go in and understand what are the best drugs for each patient. So lastly, the remaining questions basically, can we use this approach to present a mechanism for personalized medicine? And does the signaling in these xenografts actually correlate to the signaling in the human tumors? As I said, we've done a lot of work now. I don't have time to show it here. But basically, that same sort of level of complexity that we've seen in these xenografts, we now see in human tumors, where we've analyzed 21 different GBM tumors, and they basically look like 21 completely different tumors from the signaling and protein expression profile. So lastly, and most importantly, the people who do all of the work, the ERK substrate analysis was all done by a really talented grad student in my lab, Scott Carlson, who's now postdocing at Stanford. And the glioblastoma work was all done by Hannah Johnson, a postdoc in my lab, who's now looking for a position, unfortunately, specifically in London. So anyway, thanks again, and thanks to all of you for staying through the end of the day here.
I think we can take a couple questions. I have one, actually 1.5. So in your enrichment for ERK substrates in the KRAS setting, was there any enrichment for D domain versus Def domain substrates, or was all comers?
Actually, we haven't looked at that yet. It's a great question. We should.
And sort of related to that, do you have any insight into whether, any insight or any thoughts about whether maybe it would be worth doing ERK1 versus ERK2?
Yeah, not just insight and thoughts, but actually we have AS-ERK1 and AS-ERK2, and yeah, that's on the list of experiments to do.
Any other questions, or does everybody want to go to the bar? Okay, well, I'll thank all the speakers both from this session and the organizers and all of you for staying to the end. So, okay.